Hi, I'd like to welcome you to DNA Subway tutorial. In this tutorial, I'll teach you to use the blue subway line in the DNA Subway web application to do some DNA barcoding analysis. The first thing we need to do is to open a browser. I will use Firefox browser because it's the recommended browser to use for DNA Subway. Now open the Firefox browser and then go to Google. On Google, type DNA Subway and click to search. Choose the first search result and click. This shows us the DNA Subway website. The first thing we need to do is to register and create an account. Click Register and fill out all the required information. After this process, you will get your confirmation through email, and now you are able to use the DNA Subway web application. Go back to the main page and fill in your username and password to log in. Once we are logged in, we can see 5 subway lines colored differently. In this tutorial, we are mainly concerned with the blue line. So you can ignore the other colored lines for now. Click on the blue line that says Determine Sequence Relationships. This will expand the line and the 3 different steps will be shown. We will go through these steps step by step. The first step is to assemble sequence. The second step is to add sequences. The third step is to analyze sequences. But before we go through these 3 steps, we need to select our project type. Because we are doing DNA barcoding analysis, we need to select one of the barcoding options available on the DNA Subway application. As you can see, there are 4 DNA barcoding methods available. The first one is RBCL. It stands for ribulose biphosphate carboxylase large chain, and this gene is used for plant barcoding analysis. The second one is COI, and it stands for cytochrome C oxidase gene, which is used for barcoding animals. The third one is 16S, and it stands for 16S ribosomal RNA and used for bacterial identification. The last one is ITS, and it's short for internal transcribed spacer. It's used as a universal DNA barcode marker for fungi. Because I know that my samples are insects, so I'm going to choose COI as my barcoding type. Then we need to upload a DNA sequence for downstream analysis. In this tutorial, for simplicity reason, I'm going to use the sequences that are already available on DNA Subway application. To use the sample sequences, click on Select a set of sample sequences. Then click on Intro to Barcoding Bioinformatics Mosquitoes. Then we're going to name our project. And then we can also write some descriptions of our projects as well, but the description is optional. Then click on Continue. This brings us to the analysis page. We can view our sequences by clicking on the sequence viewer. You can click on the name of the sequence to further examine your sequences. I'll click on the first one. You can increase or decrease the dimensions of the sequences to take a closer look at your sequences. Once you're done, you can click X to go back to the analysis. Now we are ready for sequence trimming. Click on Sequence Trimmer. Click on Sequence Trimmer again to examine our trimmed sequences. It says your sequence have been trimmed. Now go back to the analysis. Now we are ready for pair building. Click on Pair Builder. Click on Try Auto Pairing. This pairs all the reverse and forward sequences. Then click on Save. Now we will edit our consensus sequence. Click on Consensus Builder. Click on it again to see the consensus editor. This shows us the consensus sequences for each forward and reverse sequence pair. Scroll left and right in the consensus editor window to identify what strings of nucleotides from the consensus sequence you want to trim. I will select the first sequence that says horsefly, and then I will click on the trim consensus. Here we can see some gray letters which suggests that these sequences are low quality. So I'm going to select the corresponding sequences in the consensus sequence on both ends of the sequence. Once the unwanted parts of the sequence are selected, click Trim. This will delete the unwanted sequences. Repeat trimming sequences for all of your sequences on the left. Once we are done, we have now completed the assemble sequence step in the blue line. Go back to the analysis. Now we are ready to move on to the next step, adding sequences. Click on Blast N. Click on Blast on the sequences that you want to blast. I'm going to choose the first blast. This gives us the blast results showing the accession number, details, alignment length, bit score, E-value, also known as expectancy value, and mismatches. Here we examine the blast matches for candidate identification. Clicking the species name given in the blast hit will also give additional information or photos of the listed species. One of the most important values to look at is the E-value or the expect value. E-value is the number of blast hits you expect to see by chance. The higher the E-value, the higher the probability that the alignment is by chance, and thus it's not a meaningful result. The lower the E-value, the lower the probability that the alignment is by chance. In another word, it is not random, so it is a meaningful hit. A common interpretation is that if the alignment is not due to chance, then it may be due to a biologically meaningful relationship between the two sequences. In NCBI, a usual cutoff for a good E-value is 10 to the negative 6. If desired, select the checkbox next to any hit and then click Add Blast Hits to Project. DNA Subway recommends performing a BLASTN search for all samples and saving the top 2 matches to your project for additional analysis. Depending on the project you have created, you will have access to additional sequence data that may be of interest. To add these sequences, click on Reference Data. Because we're doing a DNA barcoding project using the COY gene, samples of the COY sequence from major animal groups are provided. I will choose Common Insects and then add the reference data to our project. And it says added already. Now we have completed the add sequence step. Now we are ready for the third step to do multiple sequence alignment and build a phylogenetic tree. Click Select Data and click Select All. This selects all the user data that we processed and trimmed in step 1, and then also all the reference data and blast hits that we added in step 2. Then save selections. Then click on Muscle. This algorithm will perform multiple sequence alignment and aligns all of the sequences and creates an alignment result. Click on Muscle to view the results. Click on Trim Alignment to automatically trim the aligned sequences. Then we can view our aligned sequences. We can increase or decrease the scale to take a closer look at the aligned sequences. Then click on Flip NJ to build a neighbor joining tree. Then click on Flip ML to build maximum likelihood tree. Click on Flip NJ to examine the neighbor joining tree. Click on Flip ML to examine the maximum likelihood tree. Now we have completed the Analyze sequence step. This is the end of this tutorial. Thank you for watching.